All right. Yeah, so I want to talk about the spatial profiling of the transcriptome. So profiling the RNA while it is in the tissue. And it is, yeah, the most common localization method in biology is, of course, the microscope. And accidentally, or like coincidentally, the microscope is also the first single cell method ever, because with the microscope, the single cell has been discovered. So now it's a lot of sequencing, a lot of droplets, but I, in this lecture, try to convince you that maybe the microscope is going to be the new path for the single cell field. This is an image of the microscope setup of Robert Hooke and cells in Cork, where he first discovered the cell. So you're very familiar with this pipeline. Many of you probably did it. This is a standard single cell pipeline. You start with a tissue. You dissociate it into a single cell suspension. And then in one way or another, you compartmentalize your cells, either in droplets or in wells, or yeah, there are also other methods. And after that, you do your labeling, your cell, unique identifier, tagging, your sequencing, you do your analysis. But our tissues are not a homogeneous sack of cells. They're very structured. I have two images here of the brain and the retina, where different cells are labeled, and you see that there is a lot of inherent structure in these tissues. And even in cancer, which you would think it's more loose spatial organization, also there is a lot of spatial structure within the tumor. So spatial information has a lot of biological relevance. So it would be great if we can detect the RNA directly in the tissue, or have a way to localize or know where an RNA molecule is coming from in the original tissue, to analyze not only the transcriptome but also the spatial organization. And there's now 4 broad classes of techniques that I want to discuss with you. It's cyclic FISH, barcoded FISH, in situ sequencing, and spatial sequencing. And first, we'll focus on the first 3 methods, because they are microscopy based, and then later move on to the spatial sequencing. So how do you detect RNA in the tissue? The most common method is called in situ hybridization. In that method, you have your RNA target here in gray, and then you design a complementary probe to it, which you hybridize to the target. And this probe is labeled by something that you can visualize. A very widespread method is the chromogenic in situ hybridization. You either get this purple stain or like a brownish dap stain. Can I see some hands? Like, how many of you did this method? Quite some. There is also a variant which is fluorescent. In that case, the red star is a fluorophore, and this is called FISH, from fluorescent in situ hybridization. And nowadays, the most widely used commercial method is called RNA-seq. And you see 2 images from that method that I took from their website. So you can, on the left, you can see a lot of, it's the fluorescence for 3 different genes, I guess. And yeah, you can see tissue structures with this. So the RNA is expressed in different regions of this sample. But if you zoom in further, you can also see individual dots. And every individual dot is one RNA molecule of the corresponding gene. It works like this under the hood. So you have your RNA target, and then there is 2 probes binding. They call them Z probes, you also see. And then in a couple of steps, you hybridize more and more DNA probes to it. And this is basically done to sort of amplify the signal so that it makes it a lot easier to image. So that's great. So you can use a 20x or 40x objective, and you can see your signal without problem. And it's really nice to localize your transcripts in the tissue. Here's another 2 examples. On the left is the mouse brain, coronal section of the mouse brain, and you really see the whole overall structure. And it's easy to image a section like this. On the right, again, it's a zoom in. 3 genes in, I think the gene in white is quite lowly expressed, and that is great because then you can really quantify all the, you really count all the individual RNA molecules. However, the genes stained in the red and the green channel, they're a little bit more fuzzy. So they might have a higher density, which makes it a little bit harder to quantify. So RNA-seq is a great method to detect RNA in situ. It's very easy. I know many people that do it, they buy the proof, they work, not like antibodies. It's much more specific and much easier to do. But the downside is that, like, if you really, really want to quantify, there are better methods out there. And one of them is single molecule FISH. This is an unamplified method, which means that the dots are very small and that you can better quantify them. And it works like this. So in gray, again, it's the mRNA molecule, the RNA molecule that you're interested in. And then to every target, you design a number of probes, usually somewhere between 20 and 50 probes. And these are very short nucleotides, like 20 to 30 nucleotides long, and they're individually labeled with a fluorophore, indicated here by the colored dots. And they're designed in such a way that they tile the mRNA, they tile and they fill the whole thing up to 50 probes. If you look at that on the microscope, you can see something like this. So in blue, it's the DNA or the like DAPI or hooked signal. And the red, green, and yellow are the different gene targets. And now every dot is one single molecule of RNA. And I made a zoom in there, and now you can, it's really nice because now you can really count with how many molecules this gene is expressed in a certain cell. And for instance, if, let's say the red gene is your favorite marker for your favorite cell type, it might be easy to say that these cells with a lot of those molecules are of your cell type, for instance. Yeah, so the design principle of single molecule FISH makes it very good because the sensitivity is high and there's low false positives and low false negatives. And because there is many probes, there is a lot of redundancy. So if one of the probes is not produced or it's bad, it doesn't matter because there's still 49 other probes that bind to the same target. So you will still be able to see your signal. On the other hand, if there's one probe that is aspecifically binding to another transcript, it also doesn't really matter because it's just one probe or maybe two, but it's very hard to detect one or two fluorophores. So therefore, the single molecule FISH is it's very sensitive and very robust method. It is thought, although the actual experiment to prove it is very hard, it's thought that it has a close to 100% detection efficiency of all the RNA that there is. However, because it's a fluorescent method, there is a big downside, and that is the throughput in the number of genes. Like in any microscope setup, there is a very limited number of things that you can distinguish. If you have a normal microscope, this might be 4 or 5 colors. If you have a very fancy microscope, it might be 8. But as you all know, in your system, there might be way more cell types than 8 or way more things that you want to see than 8. So the methods that we'll discuss now are about breaking this fluorescent barrier, so to say. And the big breakthrough in this was that someone came up with the idea to repeatedly do the same do stainings on the same tissue section. So the idea is you do a first labeling in round 1, then you image that so you know where everything is that you labeled. Then you remove the signal in some way, and then your tissue section is empty again. And then you can do another round of labeling for a new set of targets. And then afterwards, you can combine all the images so you sort of aggregate the data. So then even with a very limited set of fluorophores, you can get way more data, way more throughput than the fluorophores give. That brings us to the first method, Cyclic FISH, a method that I worked on a lot. And it's conceptually quite simple because you just do repeated stainings on the same tissue section. We call our method Ouroboros single molecule FISH. And it works, like I just said, so So in the first round, you stain the targets, the transcripts of your first set of target genes. Let's say we have 4 fluorophores, so we can detect 4 genes in the first round. Then we hybridize the probes to it, and we get the signal, so we get all the dots. And after we image it, we strip the probes off. So what we do is we just melt the probes off their RNA, because they're very, very short. So basically, by just increasing the temperature, you can melt them off their target, and you can wash them away. Then, once they're washed away, you can do the next round of labeling and make an image again, and then overlay them. And now you get the composite image for all 8 genes on the same cell. Here you see a small video of how the signal builds up. Oh, it's breaking. Let's try again. So in this experiment, we did 3 genes per round. And so in every round, we add the transcripts of 3 genes. And this is a small area of the tissue that we imaged. So here's, yeah, you can see how it sort of builds up over the different rounds. But it doesn't loop. Let's go to the full, this is the full data set that we have. So again, every dot here is 1 molecule. And the color now corresponds to the gene it belongs to. This is part of the mouse cortex in the brain. And our goal was to map cell types in this area. We previously had a single cell data set of the same area, and we now wanted to map the cell types that they found spatially. Yeah, there's a couple of different broad classes of cells in the cortex. Of course, you have neurons, and they come in 2 flavors. They are excitatory or inhibitory. We put all the genes that label excitatory cells in blue shades, and all the inhibitory markers are in purple. Oligodendrocytes, which are responsible for myelinating the axons, the markers for those are in green. Astrocytes are in orange, and red are blood vessel cells. And you might already see sort of that this area might be an inhibitory cell because it's mainly filled with pink, purple dots. And this might be some excitatory cell, blue, and this might be an oligodendrocyte in green. Actually, this is, by the way, it's not an image. It's a scatter plot of the dots that we found. So that's why it looks so clean and yeah, and good. So this is the sort of the standard pipeline for these techniques. So you first do your assay, your RNA detection. Afterwards, you need to sort of segment the cells, because the end goal would be to get a single cell expression matrix like you're used to from the single cell RNA sequencing. So you need to do some sort of counterstain to find where are my cells. After you have that, so you have drew the white lines around every cell, you can now count inside each area which transcripts are there. And now you can see for every cell how many molecules of which gene are expressed. So cell number 1 has 8 molecules of the red gene, 4 of the light blue one, and 0 of the other ones. We'll talk a little bit about the cell segmentation, because this is a very hard thing to do. It's very, very complicated. Chongbin will also afterwards talk about a very good alternative for doing cell segmentation, because currently it's a very very difficult step in this pipeline. So what we did, we stained the DNA with Höchst, and we also stained the total mRNA in the cell. So as you know, every mRNA has a poly-A tail, so we just designed FISH probes against the poly-A tail. So basically it's 20 Ts with a fluorophore. If you do that, as most of the mRNAs in the cell body, you will get an image that you see on the left. And now every cell is sort of a blob that we can segment with a watershed algorithm. And then we have all the areas of the cell, and we can go count in that area. For those of you who are not so familiar with the brain, cells in the brain are very far apart, and in between there is extracellular matrix and axons and dendrites that the cells use to communicate with each other. So it's normal that they are not filled with cells like you would see in other tissues. Alternatively to this method, there is you can do the segmentation based on the nuclei, which works quite well, but there is no perfect tool for it. And if you have a more dense tissue, you could stain the membranes, or here they stain F-actin, which is close to the membranes, to use that to segment the cells. But again, Chongbin will show some more a good alternative to these methods. So after you did your segmentation, you can now look inside this area, which genes are there. So on the left, it's one cell, and the white spots is the signal, and the red crosses are the detected molecules. So now we go from an image to a cell by gene table which is the same as your single cell RNA sequencing data set. And this is nice because the images are huge. They're usually terabytes, and the gene expression matrix is usually 1 megabyte or something like that. So it's much easier to work with. And because it's very similar to the single cell, we can do very similar things with it. So we did clustering on this matrix. Here you see a heat map where the columns are the cells and the rows are the 33 genes that we labeled, and the color corresponds to their expression level. And we can find most of the cell types that we were aiming to find. And this is another representation. This is the Disney representation of the same data, and you see that it groups out quite nicely, even with so little genes. But of course, we don't only have the expression matrix. We can now go back to the tissue because we know for every cell where it is coming from. So this was our eventual goal, to make a cell type map. And now every area is a cell, and the color corresponds to one of the clusters that we found. We try to do some spatial analysis on this kind of data. So the cortex of the mouse of the brain is layered in different, it consists of different layers of different neurons, and we try to find those layers using it in a sort of unbiased way, because historically these anatomical maps have been made by people looking at morphology, looking at cell densities, and then drawing by hand the different regions. They're very good, but we thought it might be nice to do it in an unbiased way, so we made an algorithm that can find the areas, and that's what you see in the middle. So now all cells are colored by the region that they are in, and now you nicely see the layers of the cortex, sort of like a cake structure. And on the right, it's a bit a difficult plot, but we try to look at spatial relationships between all the different cell types. So what we did, we took a cell of a certain type, and then we asked, okay, what is your closest neighbor from cell type 1? What is your closest neighbor from cell type 2, 3, 4, etc.? We averaged those distances, and then we put it in this heat map to sort of see which cells are close to other cells, etc., to explore this feature. On the diagonal, it's sorted on like its self association. So the cell types that score very high for being close to themselves are the cell types that are in the layers, which makes sense because they are all packed together and they're always very close to cells of their own type. On the complete opposite, you see microglia, which are the immune cells of the brain, and these are very far from themselves. And it's also known that microglia move through the brain in a self-avoiding manner. So they have a lot of extensions, and they feel if another cell is close, and if they get close to each other, they will move away from each other. So they have a sort of self-avoiding mechanism, and that's also what we see in the data, that they are far from themselves. And another thing, which is not really surprising, but really nice for us, this red bar are the endothelial cells. So these are the cells that make up the blood vessels in the brain. And all the cell types are always close to the endothelial. not very surprising, but a very nice validation that our method is not completely weird. All right, so cyclic fish, to summarize it, it has a resolution that is diffraction limited, like it's the limit of the microscope. Depending on the wavelength, it's between 150 and 300 nanometers. That's the resolution that you get from this method. The detection efficiency, because it's based on single molecule fish, it's very high, close to 100%. The gene throughput, it's tens of genes. Basically, it's the number of fluorophores times the number of rounds that you can do. And the spatial throughput is low. It's several square millimeters in one experiment.
How many rounds can you make until the integrity starts failing?
Yeah, it's a very good question, and we don't know. So we never went to the point that it really breaks. We did experiments up to 14 rounds, and that was the point where we ran out of probes, and the tissue was still okay. We see, though, about 2 to 5% reduction in dots per round, which is pretty okay, especially if this reduction is homogeneous. Like even after round 50, a high expressed gene or a cell where it's high expressed would still be higher than a cell where it's low expressed. But yeah, like if you do with this method, if you do a 14 round experiment, it roughly takes 14 days to complete. So a 50-day experiment is not really feasible. Yeah, exactly. Yeah, it's better to do the very low first and then the higher. Yeah.
Where do you get your probes?
So there is a company that makes them. It's called Stellaris. You can buy these probes. They're about $500 to $600 per gene, and they're very good. We have a lot of them, and only one failed, and it's probably a design flaw on our side. The probes are really solid. We now make probes ourselves, and what you can also do is I will come back later to that, but a little bit cheaper method is to buy probes that have an overhanging tail. So they have like a 20 nucleotide binding region, and then there's a tail, like a flap that doesn't bind to the target. And if you make that all the same for every probe, you can buy just one fluorescent probe that binds to this overhanging tail. And this makes it a lot cheaper because then, yeah, you have a lot of unlabeled probes, which is cheap, and then you have one expensive labeled probe, but it's just one and it's not 40. Yeah.
Have you done experiments trying changing the order which you load the processes or expression over cycles?
That experiment we did not do. No, we did an experiment where we repeatedly stained exactly the same targets every other round to sort of assess the degradation. It's an epifluorescent microscope, like a wide field microscope. And the microscope is quite crucial for this because the signal is not amplified. Like per RNA target, there is only 30, 40, 50 fluorophores. So the signal is very weak. So you need a very high magnification and a very sensitive camera to do it. And the confocal does not work for this method, or it doesn't work so well because confocal is point scanning. And because the signal is so low, you would need a lot of exposure time per pixel, basically. So it's best to go for an epifluorescent wide field microscope.
So when you start experiment, is it important that you make everything, like the whole cell, that's low and using and changing things?
Yeah. The important thing is that for this method, or for most of these cycling methods, that you always image exactly the same region. Because if you image multiple regions, you don't aggregate the same data on the same cells. So it's very important that the microscope stays on and no one calibrates it during during the experiment, because if you recalibrate it, it's lost. We had, it was a power outage, microscope lost its calibration, and we could throw away the image, the whole experiment. So it's in the, like the chemistry is easy. If you can pipe it, you can do it, but the imaging is tricky. Or you need a specialized or maybe even dedicated system to do it. Yeah.
So it means that you use the laser immersion objective. How it's going on? So you have the slide, right? And you slide there, and then like you said, and it's like not the water, right? So how you put the microscope? You're not covered the slide, right?
No, so the sample is in a flow cell, so we can put in liquids, and it's on a cover, like the tissue is on a cover slip that we can access with the objective. And in the experiment I just showed, we did everything on the bench, and then in the evening went to the microscope, put it on the microscope, let it image the whole night, and then I took it off again and did the next round in the lab, and then went back to the microscope again. And it's, yeah, we had a special frame. I don't have the image here. We have a special frame, which is basically a 96 well plate format, and then you can place it in the stage. And because there's the spring lever, it will always center it in the same position. And we imaged with the displacement of a few micron per round. Like it's quite stable to just take it out and put it in. We now have a system where the sample stays on the microscope, which, yeah, doesn't really add so, like, data is equally good, but it's a little bit easier because it costs a little bit less time for us to do the experiment. Yeah, so this is a very good question. So we use 10 micron sections, but it depends very much on the imaging, how deep you can go. The staining works in 3D, but just the imaging is very tough because it's high magnification, high numerical aperture objective. So the focal depth is very, or like the working distance is very close to the objective. So it's very hard to image really deep, but people have been doing 100 to 300 micrometer. And yeah, you might need to do some clearing. You need a good immersion oil and good immersion, imaging buffer to enable this, but it's definitely possible. Yeah. All right. Should we break now, or can do a little bit more? Yeah, maybe let's break now for, is it 15 minutes or? Yeah, for me it doesn't matter, but okay. Yeah. Okay.
Okay.
Yeah.
Kaikkien rekisteröityjen tutkimukset, kun hakee resursseja, niin se kannattaa tehdä sen ryhmänjohtajan taas lukuja, jotka tavallaan voi tuoda teknisenä ryhmänjohtajana nähdessä. Täältä sivulta täältä, kun Kansan projektissa nähdään, miten systeemi menee. Miten haetaan projekti ja sitten haetaan nämä computing services, mitä ottaa muun muassa tuon C-puolen, että on mitä yhtiössä on se todistua siellä.
Onko tämä semmoinen, että pitääkö sinun joku asia aloittaa ennen?
Välttämättä ei näe pystyy tekemään sen kohtuuden. Mutta sitten se, mitä teidän pitää tehdä, on yhden teidän kannalta. Ja sen pitää sitä nostaa taloudesta, se laajennetaan 20 se on tehty sitä läpi niitä. Se vaatii sen, että siellä ei
Yeah.
All right, let's continue. So I just talked about the cyclic single molecule fish, and we'll now move on to the next method of the four, which is barcoded fish. And barcoded fish is rests on the idea that you can not only do multiple cycles, but you can also stain the same RNA molecule multiple times. And yeah, they build a barcode on the transcripts to identify even a larger number of proteins. So what they do is, like in these methods, you hybridize your probes in the first round, you image it, then you strip them off, and then you hybridize to the same molecule again in the second round. So then you get, you measure the same molecule twice. And the smart thing is that now you can make a very smart barcode on the targets to increase the number of targets with only a very limited number of fluorophores. So here it's a very simple example. We have RNA A to I, and we're going to do 2 rounds. And we make we predetermine a barcode for every, like a color code for every gene. So RNA A will be stained red in the first round and red again in the second round. RNA F will be green in the first round, and then in the second round it will be blue. So now with only 3 fluorophores in 2 rounds, we can label all these transcripts, which in a cyclic method would be much less. So this is what it would look like. So in round 1, you stain all your molecules. Some of them are red, some of them are blue, some of them are green. You have no clue what they are yet until you do the second round. So in the second round, again, they light up in a different color. If you then overlay these images, you can sort of reconstruct or like find the barcode. So now on the left, you see the color from the first round. On the right, slight right, you see the color of the second round. And now you have the barcode, and with this and the predetermined library, you can sort of identify the dots. So very important is that in round 1, you see these 3 molecules that are labeled in red. And at this point, it can be still RNA A, B, or C. And then with the second round, you suddenly resolve them, and you find all the identities of all the detected molecules. And this was first proposed by Longkai in 2013. This is an image from their paper demonstrating this. So you see these, and in the bottom you see the dots changing color. And that is part of their barcodes. And the fantastic thing is that this barcoding scheme scales fantastically. It's the number of fluorophores that you have times the number of rounds. So with just 4 fluorophores and 7 rounds, you can do, I put the comma wrong. No, correct. It's 16,000 targets that you can do if you do one cycle more, you can resolve the human transcriptome 3 times over. So, so the barcoding capacity is huge. They started a little bit smaller. The method from the lab of Lonka is called SeqFISH, and there they make these barcodes on the RNA, and with also their goals to resolve cell types in the brain. They use a slightly different labeling technique. This is called hybridization chain reaction. It does not label the RNA with just one fluorophore per probe, but it will build sort of a long stretch of probes with fluorophores. Basically, how it works is that first you hybridize a probe that has a gene-specific, like our target-specific region, and an overhanging flap. And this overhang is the initiator for the hybridization chain reaction. So in the staining solution, there's these hairpins with a fluorophore, and they can be broken open by the initiator. And when they break open, they extend, and then the next one comes, and the next one comes, and the next one comes. And so it builds up a lot of, a large structure with a lot of fluorophores. So these dots are very, very easy to image, which makes it a lot more feasible. And then to remove this, they use DNAs, because the probes are DNA, the RNA is not affected by the DNAs. So they use the DNAs to remove these probes, and then they can do the second round. And then they do 100 genes barcoded to resolve them. And they also have 25 genes that they do in a more serial, like cyclic FISH manner. And with that, they could find dots in the brain. This is a small piece of what they imaged. And in the bottom, you can see the examples of the dots changing color over the different rounds. And then they can identify them afterwards. And what they could do, they could then, after segmenting their cells, they could now, in this image, the cells are colored to the clusters that they found of the data. This is the dentate gyrus in the mouse brain. However, there are 2 big challenges with barcoded methods. That are first is that there can be errors in the barcode. Like if you label the same molecule multiple times, you increase the chance that something goes wrong. And secondly, the optical density is an issue, and I will come back later to that. So first look at the errors. So here's the same example, but now something is going wrong. So let's look at the top red dot with the arrow. Let's say it's stained perfectly nice in the first round, but for some reason in the second round it is not there. So this would mean that if we're gonna identify it, we can't, because we don't know what it exactly is. In the bottom arrow, there are 2 molecules that are very close to each other. One is red and one is green in the second round. And by alignment errors or by some chance, we think that our original dot is green instead of red, which gives a wrong barcode and like a misidentification of the dot. So instead of thinking that it's RNA A, we now think it's RNA B. And to deal with this kind of errors, Xiaowei Wang came up with a method called MERFISH, which is a multiplexed error robust FISH. What they do, they have a more sparser barcode with some nice tricks. So what they do for every target, they design a binary code, like 1s and 0s. And then they do the labeling, but these barcodes, they are designed in such a way that they cannot easily mutate into another one. And what they use is, they use the Hamming codes, which are traditionally used for telecommunication to make sure that your messages, your email arrive safely at the other end without errors. And so let's consider the top example, barcode 1, barcode 2. So these are very similar. The only thing is that the first bit is different. So the barcode 1, it's a 1, in barcode 2, it's a 0. So if by reading the barcode, something goes wrong in the first bit, we read a 0 instead of a 1, we will misidentify the target molecule. We would think it's belonging to barcode 2 instead of barcode 1. So what you can do is you can pick barcodes that are very more separated from each other. So these barcodes are very similar, just one bit needs to flip to cause a misidentification. So the smart thing they do is use these Hamming codes with a distance of 4, which means that 4 bits need to flip to be misidentified. And it's pretty rare that you have 4 errors in a barcode, so then you're more safe. Yeah, you also see it in the bottom example that this is an example of Hamming distance 4. So 4 bits need to flip to become the closest next barcode. If there is 1 or 2 bits going wrong, you can if you know 1 bit is flipping, you can correct the error. 1 or 2, no, if 1 or 2 are wrong, you can correct the error. If it's 3 bits, then it becomes more challenging. So, but this is a very great way to deal with these errors. And this is a little bit about the staining strategy of what they do. So first they hybridize probes to the target with the same sort of overhanging tails that I talked about before. And in these overhanging tails, the barcode is encoded, sort of. And then they have fluorescent probes that they hybridize to these overhangs, and they will only hybridize if there should be a 1 or a positive in the barcode. And they have the current method has a nice strategy of stripping the signal off. So our method uses high temperature, or basically like lower melting temperature with formaldehyde to strip the probes off their targets. Longkai's method was using the DNAs to digest them, but in MERFISH they are now using probes with a thiol group, like sulfur-sulfur group, between the DNA and the fluorophore, and they can reduce this bond with a reducing agent, and then the probe stays, but the fluorophore goes away. So then it's empty again, and this is really quick. Like within minutes, you strip your signal off, and then you can go to the next round of labeling. Yeah, and on the right you see sort of their encoding scheme. So they have a barcode, let's say with 8 bits. In round 1, the red fluorophore will encode for bit number 1, the yellow for bit number 2, etc. And then in round 2, the same fluorophores are used, but for another part of the barcode. So in round 2, it's bit number 5 is red, and bit number 6 is going to be the yellow fluorophore. And that's how they build up their barcodes. Here we can see an example of this, of how they do it. And I wanted to focus on this dot here. So in the first round, there is nothing, so it's a 0. And in the second round, there's a dot, so it's a 1. Third round, it's not there. Fourth, it's there. Fifth, it's there. Then it's empty, I think, until number 13. And I think it's, if I look correctly, it should be this one. So it should be number 6. This is the barcode. So it's 0 in the first, positive in the second, and then you can identify the gene belonging to this barcode. They have quite some publications, and I think this method is pretty mature, as in they can routinely do this with high throughput. Here they were looking at the thalamus of the brain. They did 135 genes with their barcoding strategy. They did multiple sections from posterior to anterior in the brain. They did behavioral studies as well. And they could quite nicely find the cell type belonging to each cell they found. And it also gives the same sort of cell type maps plus everything else you can do with it. Another very nice feature that they introduced with the MERFISH protocol is gel embedding. So as I said, the signal coming from these methods can be very low. It can be very hard to image. And like tissue background can be an issue, especially like in the green channels. Maybe some of you are familiar with that. So what they did is they cast a gel, like acrylamide gel, in the tissue, cross-linked their RNA to this gel, and then when the RNA is fixed in the gel, they digest the proteins. So they digest most of the cells out using proteinase K. And that leaves the gel and the RNA. So if you then do your staining, you get a lot less on the tissue. So it removes the autofluorescence from the tissue, and it also removes the chance that the probes are binding to proteins inside the tissue. So it's a really nice feature that they added. Yes. Now come back to the optical space, which is a big limiting factor for barcoded methods. Here you see an image or a fish image, and most of the dots are pretty nicely resolved. They're alone and they're quantifiable, but the ones with the arrow, I don't know if that's 2 dots or 3 dots or 1. It's very hard to notice what's going on. And this is because light is diffraction limited, so there is a minimum size that you can see. Like dots don't get smaller than the diffraction limit, unless you go to super resolution microscopy, but that is very, very slow in terms of spatial throughput. We did some simulations. So on the bottom right, you see cells that we made. So these are artificial cells, and we start filling them with dots, and then we use our detection criteria of how far dots need to be based on the diffraction limit, how many we can actually detect. So all the dots that are green are correctly detectable, and all the ones that are red are too close to each other to be detected. And we run this simulation from 0 to 1,000 dots. We see that there is sort of a peak around 125 resolvable objects in an average brain cell. And after that, they get so close to each other that that their number decreases. For us, we lose data. Like if you do a single round of single molecule FISH, you lose data. But if you do a barcoded method, you might lose the whole. This might happen multiple times because you measure the molecule multiple times, and you might lose more because of that. So there's 2 solutions to this problem, and one is expansion microscopy. Maybe some of you heard of it. This is basically you physically enlarge the tissue. So you basically, if 2 dots are very close to each other, but by expanding the tissue, you will pull apart those dots, and then they will be separated, and you can detect them individually. It's a very nice method. It of course also will increase the imaging time a little bit, but it's a nice method to really resolve it.
First staining and then
expansion. But the tricky thing is that for every imaging cycle you need to expand again, because the expansion is based on salt concentration. So the hybridization of the probes is done at like physiological or double physiological salt concentration, and then it's expanded by putting the sample in water. So it's a little bit tricky to do the staining, expand it, image it, and then go back. There are some issues to align your images afterwards, but it is possible. So the barcodes that I had put in the example here are dense barcodes. So which means that every gene is always labeled in every round. So all the genes are always labeled. In the MERFISH, where they had the binary code, there were zeros in there, which means that the transcript is not labeled. And this is a very nice way to deal with the optical space. So on the left, again, it's a dense barcode. So every molecule is always labeled. And you will see that, basically it's one blob, and it's hard to say what is an individual molecule. However, if you have a sparse barcode and you stain only certain dots in certain rounds, and not so many, you really dilute the dots, so the crowding becomes much less. You need to do a little bit more rounds to stain the same thing, but you're actually able to resolve what you're looking for. And this is what they did, of course, what they did in MERFISH, but also what they did in the latest publication from the Lonkai lab, which is now called SeqFISH Plus. And in this method, they have a quite sparse barcoding scheme, and they resolve up to 10,000 genes in a tissue section and in cell-cultured cells. So with this paper, they get really close to full transcriptome and gets very close to competing with single cell RNA sequencing. Here's an example of their data. So here they zoom in on one cell in the brain, and you see all the dots corresponding to an individual RNA molecule. Yeah, you see how dense it is, but because of the very sparse labeling, they're able to resolve this. And in the bottom, there's a cell type map or like cluster map what they found of these imaged cells. So that's the barcoded single molecule FISH methods. So the resolution is still diffraction limited. The detection efficiency is high, but a little bit lower because errors might play a role. The gene throughput, since 2 months ago, it's now up to 10,000. And I think there is not much limitation to go full transcriptome if they want. The spatial throughput is low, several square millimeters. And that brings us to the third microscopy method, which is in situ sequencing, or like sequencing in situ based. So this is another method to identify RNA molecules in the tissue, but it uses a slightly different approach by sequencing the molecules while they are still in the tissue, instead of putting it on an Illumina sequencer, for instance. As you might know from an Illumina sequencer, what you do is like you have your library that you want to sequence. You load it on the Illumina sequencer, and it will do this bridge PCR to generate sort of spots with a lot of copies of the same molecule. And that is done to sort of amplify the signal if you are going to sequence it. And similarly, if you want to sequence a molecule in situ, you also need to sort of amplify the thing that you are going to sequence. So these methods are all based on rolling circle amplification, which basically uses which in this method called in situ sequencing uses a padlock probe, which is the blue part. So you have a probe which binds to the mRNA, and the ends get hybridized very close to each other, and then you ligate these to each other. So then you have a circle of DNA. Then you use polymerase to, basically amplify, like it will go in circles and produce many copies of the same circle, which you can then use to do the sequencing. So this method works with sequencing by ligation. So you have many copies of the same sequence. And in this method, it's targeted. So you design your padlock probe targeted to a certain mRNA. And then the part that you want to sequence is a specific barcode. In this case, it's 4 bases. And then you have your sequencing probes that you put on. And one of the, so the first black part is fixed. So that's the part that you want to sequence. Then there is the colored nucleotide, which is specific. So red stands for an A in this case. And then the other nucleotides, the ends are random sequences. So there's always probe binding, but it will be specific for the A. So if your dot lights up in red, you know that the first nucleotide is going to be an A. Then you melt it off just by stripping it, and then you move on to the second nucleotide. And now everything are ends except the second one, which is specific. And if it's a C, it will be green. And in this way, if you cycle this a couple of times, you can sequence the barcode that you put in there. And the scaling is quite nice. You could sort of see that the nucleotides are the fluorophores, so it's 4 times the number of rounds, minus something if you want to do barcoding, or like, if you want to do some error correction in the barcode as well. And this is their most recent publication, now in bioRxiv, where they focused on finding interneuron cell types in the hippocampus of the mouse. On the left, you see the 5 rounds of sequencing, and you see that the dots are changing color according to their sequence. And because this signal is very amplified because of the rolling circle amplification, it's quite easy to image, and they can image with a 20x objective, and they can easily image the whole mouse brain section like this. Yeah, one thing I need to note is that they first do a reverse transcription. I forgot to mention that. So they first reverse transcribe the RNA, then put the padlock probe on, and then do the rolling circle amplification. In this process, there is some loss, and it's not as sensitive as the single molecule FISH. It's a bit lower than that. There's an alternative method which is very similar, but has a one very different step. It's called FISSEQ. It also first reverse transcribes the mRNA, but then instead of using a padlock probe, it circulizes the cDNA itself. So, and then if you do rolling circle amplification, you get the exact molecule. And this is great because it's untargeted. So it basically, it can target any RNA. So it's a very unbiased way of doing the same things. And then they use the commercial solid sequencing to read out their barcode, or to read out their sequence. And so they really get, unbiased sequences from their targets. The thing is, it's a little bit inefficient, probably because the parts of the mRNA can be very far apart. And because it's unbiased, it's also hard to exclude ribosomal RNA. So a lot of reads can come from ribosomal RNAs, which are very abundant in the cell compared to mRNA. More recently, there's another method based on the in situ sequencing, which is called StarMap, and they have a slightly different approach to making this rolling circle amplification. So they don't do reverse transcription, which is great because it's very inefficient. So they have a probe, which is like identifies the gene like this, and then they use another probe, which they call the snail probe, to bring the end of the first probe together. They ligate this together and then do the rolling circle amplification. What they also do is you see that there are amine groups on the amplicon, and this they use to embed the amplicon into a gel. They digest the tissue, so then they have just the amplicon plus in the gel without any interference from proteins or other things that are in the cells. And then they also do sequencing by ligation to find their barcodes. And with this, they can resolve. They showed up to 1,000 genes barcoded, and also in 3D. They use the gel to go to more 3D sections or 3D structures. Yeah, here's an image of the slice, what they do. So they have 100 micron, in this example, there's 100 micron thick tissue slab where they do their assay on. So to conclude the sequencing in situ, the resolution it can be diffraction limited, but it's but probably the amplicons are a bit larger. So the size, the resolution is half to 1 micrometer. The detection efficiency can be very low. In the case of FISSEQ, it can be as low as 0.1%. And in the StarMap, it can be maybe 50%, but be very cautious with this. Because we don't really know, they didn't really do a good comparison with single molecule FISH. So we don't really know exactly how efficient it is. The gene throughput, of course, for FISSEQ, which is unbiased, can be the whole transcriptome, which you will just have very little reads. But so I put like somewhere between 10 and 1,000 here to sort of have a ballpark range. And the spatial throughput can be very good, from several square millimeters to several square centimeters, because it's amplified. And of course, you can also, because it's amplified, you can also go 3D. So it could be a few cubic millimeters, or maybe a cubic, maybe a cubic centimeter, but it will be tough to image. And then the last spatial method, which is spatial sequencing. This is not a microscopy-based method. It's a very different approach to get spatial information from RNA. And instead of capturing the spatial information in light or in an XY position, they capture the spatial information into DNA, into a DNA barcode. So what they have is they have a surface, and the surface is coated with capture molecules, very similar to what they use in the single cell RNA sequencing. And these capture probes, they have, of course, a poly T, which can hybridize to the poly A of the mRNA, but it also has a spatial barcode. Here it's shown in yellow. And then they have spots on the glass slide, and each spot has a unique spatial barcode. So all the capture probes in that spot have the same spatial barcode. And then they put, you see it in the bottom, like it's the different IDs or the different spots with the different spatial barcodes. Then they put the tissue on top, permeabilize it so that the RNA can diffuse out and be captured by the underlying capture probes. And then they remove the tissue, do reverse transcription, and now the barcode, the spatial barcode, is incorporated into the reverse transcribed molecule, into the cDNA. So in that way, they link the mRNA to a spatial barcode, and if you sequence that, you can see, okay, this RNA is coming from this spot. So in the original publication, the array looks a little bit like this. So every spot has one unique spatial barcode. They are 100 micron in diameter, and they are spaced, their edges are 100 micron from each other. So they say that in the brain, each spot contains about 30 to 40 cells, but it depends on your tissue, of course, how many it actually is. Yeah, this I already said. Yeah, this is an example of their data. So now you see on the right, you see two tissue sections of the olfactory bulb in the brain. And every colored dot is one of the spots, and they clustered the expression profiles of these spots, and they can basically find different regions in this brain area. And yeah, it quite well matches the morphology of the tissue. But the resolution in at least the original publication is very crude, 100 micrometer dots, quite far apart. So it's not single cell. It's not a single cell method. But recently, one publication in Science and one bioRxiv showed the same principle, but with a much higher spatial resolution. And so what they do is they use beads on an array to encode the spatial information. So they start with a randomly barcoded beads. So beads, they have the colored line is the spatial, is the barcode, and then they have a poly T capture site. Then they deposit these barcoded beads on an array, so they're spatially restricted now. They're on the surface, and they use some sort of sequencing or some sort of FISH-based method to read out the barcode of every bead. So now they know where every bead is in space. Afterwards, they place the tissue on top, let the RNA diffuse, bind to the beads, and then they do the sequencing of the barcode plus the captured mRNA. This is the published one. It's called SlideSeq. So starting in the left, the top corner, you have these beads. They are packed on the surface, and they use in situ indexing, like they sequence the barcode to resolve the location of these dots. So now they know for each dot, for each bead, where it is, where it's located. Then, like I said, they put the tissue section on top. They let the RNA diffuse out, get captured by the beads, and then they can transfer it to the tube, They do reverse transcription, tissue digestion, and then they can make the library out of these beads. So, and then by combining the read of the RNA and the location of the known location of the bead, they can reconstruct the sequencing results spatially. So here's a couple of examples that they made in the brain, where you can really nicely see structures. So every dot, I guess, here is a single bead, and they clustered them based on their transcription profile. And they quite nicely see all the structures that are known to be in the brain. Noteworthy to say is that these beads are 10 micron in diameter, so they're roughly the same size as a cell. But yeah, how a cell is placed on top of these beads is, of course, can be a bit under the influence of chance. Very similar approach, now on Barcoded, uses also a bead array which are coded with these capture arms with a spatial barcode and a poly-T. It's a very similar process as I described before. The only big difference is that these beads are 2 micron in diameter. So they're much smaller than a cell. So they should have a much higher spatial resolution. So that brings me to the end of the spatial sequencing. So the resolution is somewhere between 2 micron and 100 micron, depending on your spot size or your bead size. The detection efficiency is quite low. So estimated to be somewhere between 0.1 and 1%, maybe a little bit more. But the gene throughput is super high because you can sequence whatever. You can sequence the whole transcriptome, but because you have the original mRNA there, you can do a lot of things that are not possible with FISH, like single nucleotide polymorphisms or intron-extron boundaries. Like all these things you can now sequence, which is a huge advantage. Also great is that it's based on sequencing, and sequencing just works. FISH, like I said, with the microscopy can still be a little bit tricky, but Illumina sequencing is a pretty solid technique, so you can just sequence, which is nice. The spatial throughput is great. Can do several square centimeters. It depends a bit on how much money you want to spend on the sequencing, but in principle, you can scale. This method could scale very, very nicely to large areas, provided that you want to pay for the sequencing. And that is the end of my part. So discuss these 4 broad classes of spatial RNA detection methods, and I would be happy to answer any questions if you have. Yeah, yeah. It depends on the expression level, but yeah, a few percent of the molecules that are in the tissue section actually get captured by the slide. Yeah, 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 because they just stay there, and it's easy to stain them. Yeah. All right, then Shungbin.